Gene-Space Glaucoma Trials, April 2026. Emerging gene and cell therapies hold promise for glaucoma, a disease that slowly destroys the retinal ganglion cells, RGCs, the nerve cells sending vision signals to the brain, and impedes the eye's natural drainage of fluid, the aqueous outflow pathways. These next-generation treatments aim either to protect or regenerate RGCs, neuroprotection, or to improve outflow tissue function and lower intraocular pressure. In April 2026, several first-in-human trials will begin for such approaches. Below, we summarize their main features, vectors, molecular targets, dosing plans, and immune safety measures, as well as how they are delivered and controlled. We also note the ethical issues of sham controls and the required long-term safety monitoring. Gene therapy for RGC neuroprotection. Some trials deliver genes encoding neuroprotective factors into the eye to help RGCs survive. For example, one approach uses a harmless viral vector, often an adeno-associated virus, AAV, to carry the gene for ciliary neurotrophic factor, CNTF, or brain-derived neurotrophic factor, BDNF, into retinal cells. These proteins act like growth factors to keep RGCs healthy. Indeed, laboratory studies report that factors such as BDNF and glial cell-derived neurotrophic factor, GDNF, can greatly improve RGC survival. In an upcoming Phase 1 trial, for instance, patients will receive an intravitreal into the gel of the eye injection of an AAV vector carrying the human CNTF gene. The trial is dose escalating. Each group of patients will get a higher viral dose to find the safe and active range, typical Phase 1 design. Blood and eye exams will regularly check for immune reactions. For example, measuring if the body makes antibodies, binding or neutralizing, against the viral capsid or the new gene product. Many ocular gene trials also use short courses of corticosteroid eye drops around the time of injection to blunt inflammation. Another putative gene therapy targets the neuronal degeneration process itself. For instance, trials may deliver genetic braking tools like short hairpin RNA or CRISPR nucleases to suppress harmful signals in RGCs. One example in animal studies used an AAV-delivered Cas9 gene editing system to knock out the Wallerian degeneration pathway, which causes axons to die after injury. In mice, such treatments kept RGC axons more intact. Key points: gene therapies for RGCs typically use intravitreal or subretinal injections, small eye surgery akin to injections for macular degeneration, and monitor vision function by elective tests, imaging, visual field, etc. over time. Because gene expression is long-lasting, trials plan extended follow-up. FDA guidance, for example, calls for up to 15 years of post-treatment monitoring in gene therapy trials, focusing on late adverse events like tumor development. A registry of treated patients may also be kept to flag any rare issues. Gene therapy for aqueous outflow. IOP lowering. Other April 2026 trials aim at lowering eye pressure by improving the drainage of fluid. These target the trabecular meshwork and Schlem's canal, the tissues in the iridocorneal angle that normally let aqueous fluid exit the eye. One promising strategy is gene editing. For example, an AAV vector carrying CRISPR-Cas9 components can be injected shared from the anterior chamber so it transduces trabecular cells. Preclinical studies have shown that knocking out certain genes can reduce intraocular pressure. For instance, in mouse glaucoma models, an AAV CRISPR targeting the water channel gene AQP1 significantly lowered eye pressure and prevented RGC loss. Similarly, targeting the glaucoma gene myocilin with Cas9 in mice removed the harmful protein and produced a sustained IOP drop. Other trials may use AAV to deliver enzymes, like matrix metalloproteinases, or inhibitors of scarring to the trabecular meshwork, aiming to boost natural outflow. In every case, the trial protocol will describe a dose escalation plan, starting with a low vector dose in the first cohort, then higher doses in subsequent cohorts, to find a safe dose. Throughout, researchers will test blood for antibodies to the vector and transgene as a measure of immunogenicity, and will grade any eye inflammation via exam and imaging. Because the eye's anterior chamber is relatively immune-privileged, severe reactions are uncommon, but monitoring for uveitis or other inflammation is standard. Surgical delivery. Outflow targeting gene therapies are usually given by a small injection into the eye's drainage angle area. This can be done by a surgeon through a tiny corneal incision, similar to glaucoma drainage device placement. or as a suprachoroidal injection. The delivery must precisely reach the trabecular meshwork Schlem's canal cells. Good surgical technique and imaging, often optical coherence tomography during surgery, help ensure the vector is where intended. Cell-based therapies. In parallel, some trials will test cell therapies for RGC support or outflow tissue repair. For RGCs, one example is an encapsulated cell implant that produces CNTF. This device, an inchworm-sized capsule placed inside the eye, contains modified human nerve cells that steadily release CNTF. The implant is placed via vitrectomy surgery and stays in the vitreous chamber. Like NT501 implants studied previously, it provides continuous neurotrophic support without repeated injections. Phase 1-2 results from prior studies, not in glaucoma but in macular conditions, showed safety and slow release of CNTF. The April 2026 trial will further test whether dual implantation or higher CNTF output can protect glaucoma eyes. Patients will have regular eye exams, imaging, and visual function tests to spot any inflammation or tissue reaction to the device. Because the implant cells are contained, systemic exposure is minimal, but as with all gene cell therapies, monitoring includes checking for antibodies to any cell-derived proteins. For aqueous outflow, a key approach is transplanting or injecting stem cells into the trabecular meshwork to regenerate its filtering function. For example, an autologous patient-derived trabecular meshwork stem cell or mesenchymal stem cell (MSC) could be injected into the anterior chamber. Using the patient's own cells greatly reduces rejection risk. NYU researchers have proposed exactly this, prioritizing autologous MSCs for TM repair. Early-stage trials will test safety. Dose escalation might mean injecting low cell numbers first, then higher amounts. Patients' eyes will be checked for unwanted growths or inflammation. If allogeneic cells are used from donors, immune suppression, like short-term steroid, may be applied. Notably, prior ocular stem cell trials, e.g. retinal pigment epithelium transplants, saw only mild immune reactions, which were controlled by local steroids. No tumors or serious adverse events are expected if cells are well characterized. Even so, imaging, OCT, and vision tests track any side effects. Ethical controls. Sham and delayed treatment. An important design issue is the control group. In classic drug trials, a pill placebo is given, but for in-eye interventions, this is hard. A sham surgery, a fake injection without actual gene cell delivery, would give ideal blinding, but is fraught ethically because it exposes patients to procedural risk without benefit. Experts warn that sham surgery trials demand elaborate justification and safeguards. In practice, glaucoma trials often use alternative controls. For example, they might compare treated patients to those on standard medication alone or use a delayed treatment control, patients assigned to wait 6 to 12 months, then receive the therapy. This way, all patients ultimately get the experimental therapy, and short-term outcomes can be compared before the delayed group is treated. Such designs balance rigor and ethics, acknowledging glaucoma leads to irreversible vision loss if left untreated. Any use of sham or delay must be approved by ethics boards and clearly explained in consent forms. Long-term safety and follow-up. Because gene and cell therapies are potentially permanent or long-lasting, regulators require extended safety monitoring. The FDA's guidance for gene therapy products, for example, mandates up to 15 years of follow-up for patients, with regular check-ins on injection sites and whole-body health. Key data collected over time include ocular exams to catch delayed inflammation or degeneration and general health screens to detect any malignancy related to the vector. Patients may also be enrolled in a registry so outcomes can be pooled and analyzed across many years. For cell therapies, long-term follow-up, often 5 to 10 years, is also advised to watch for late adverse effects. In practice, trial protocols specify visits well beyond the end of dose escalation. Annual eye exams and vision tests, plus any blood tests needed, continue for years. This ensures a safety net. If any rare issue, like a viral vector causing genome changes, emerges, it will be caught. In summary, the early April 2026 trials of gene-modulating or cell-based glaucoma therapies will involve carefully chosen viral carriers or cells, well-defined molecular targets, e.g. CNTF, BDNF, MYOC, AQP1, extracellular matrix factors, and stepwise dose escalation. Immunogenicity will be monitored with blood antibody assays and eye inflammation grading. Delivery will be surgical, intravitreal or intracameral injections, or implants under sterile conditions. Controls will favor delayed or standard care arms rather than risky sham surgery. And all subjects will enter multi-year safety follow-up, often in registries, to ensure long-term eye health. These measures follow current recommendations for gene and cell-based trials and aim to maximize patient safety while testing these innovative glaucoma treatments. Sources: recent reviews and guidance documents on ocular gene cell therapy are used to outline these trials. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.